Hello my dear students and my dear friends. Today we will be talking about translocation. Before we start with the video, those who are watching my channel for the very first time, do not forget to hit the subscribe button, share the video with your friends, and don't forget to like the video. But at the same time, if you have not liked my video, if you have some really critical feedback for me, do not forget to write it below in the comment section. That will help me in improving for my next set of videos. Anyways, let's begin. So first, let's understand what is the meaning of translocation. Well, translocation is a process through which a plant body transports all its assimilates from one place to another place. Now, what do you mean by the word assimilates? Assimilates are any kind of organic molecules which are present inside plant body, and they are required by each and every cell. So you think about the glucose, you think about the hormones, they have their own different functions, right? So all the cells deserve to have all those kind of assimilates. Now you have to make sure that those assimilates get transported from their place to the place where they are deserved to be. So that brings me to the next point. Source is a place of the plant body where you can prepare all those assimilate. Rather, they are being utilized there. And from the source, you have to make sure that all those assimilates transport, or they reach to the place known as sink. Sink is a place where those assimilates will be utilized rather than produced, right? So in due course of time, we will be talking about all those keywords. As you can see, I've written different types of keywords. So you talk about proton gradient, apoplast, implast. All of these examples will be taken into consideration when we will discuss the translocation as a process in detail. Now let's begin with that. All right. So I have tried to make a very schematic brief diagram for your understanding. So, well, this is a plant body which is doing photosynthesis in the presence of sunlight. Now that glucose need to be sent to different corners of the plant body. It can be upward movement, it can be even downward movement. So that translocation of the glucose is a classic example of bidirectional transport. But in some of the textbook, you may get the word as multidirectional transport, which is also correct. Anyways, let's get into the core of the understanding. Now basically what happens? Once you prepare the glucose, which is said to be a monosaccharide in the plant body, that glucose will be converted into sucrose, which is a disaccharide. Once the conversion is done, that sucrose will be transported from the source to the sink. So as far as this picture is concerned, your source is the kitchen of the leaf. I should say kitchen of the plant, that is a leaf. From here, the food need to be transported to different corners of the body, that is the sink, where it will be utilized more. Here they are produced more. So this is how you can define as far as definition kind of question is concerned in the exam. So if you make a cross section here, this stem, if you make a cross section here, and if you magnify that part, you will see kind of the cells which are present. So like I've told you already, translocation is a mechanism which will be done exclusively by phloem. Now phloem is a type of complex tissue which is dead in nature. Which is living in nature. Phloem is a kind of complex plant tissue which is living in nature. On the other hand, xylem is another type of the vascular tissue which is dead in nature. You know the difference between xylem and phloem. Xylem and phloem, they're all complex structured tissue. That means they're made up of 4 different types of cell. If you consider xylem, out of the 4 different cells, 3 cells are dead. So majority wins. So xylem is considered to be a dead tissue. On the other hand, if you consider phloem, which is again made up of 4 different types of cells, 3 cells are living in nature, as a result of which it's considered to be a living tissue, right? Now I have taken two different type of cells out of the four, like 4 cells of the phloem, because these are the 2 cells which will be actively participating in the translocation. So if you see, this is a kind of cell which are arranged one after another, and they are a kind of cell to cell contact is there. So you can see these are the cell to cell connectivity. So overall you can see 1, 2, 3. So I have drawn 3 cells. These cells are known as sieve tube element or sieve cells. SIEVE, sieve cells or sieve tube element. If you see these cell to cell connectivity, they have got a number of perforations. So this is not totally a solid structure, rather you have a number of pores in between. These are known as sieve pores, and that's why this plate is known as sieve plate, right? Now what is the function of the sieve plate? We'll be understanding very soon. Adjacent to this cell, try to understand one thing. There are multiple cells of this type. So for this, I have taken one for each of the, for a better understanding, right? One for each. So here, as you see, adjacent to the sieve tube element, there will be one cell which is said to be companion cell. So this is your companion cell. Now try to understand, both of these cells are living, but these sieve tube element, most of the cell organelles are missing, especially the nucleus, and cytoplasm is also very less. Why? Just to give more space for the assimilates to get accommodated. You remember the reference of red blood cell? Why red blood cell, I should say mature red blood cell, lacks nucleus and all the cell organelles? Right, it's kind of more similar function. On the other hand, you have this full-fledged cell which is known as companion cells, and this cell has a lot of mitochondria. It has got its own nucleus and also the cytoplasm along with the other different types of cell organelle. Now try to understand, this cell lacks nucleus. Now as you know, nucleus is said to be the regulatory center of a cell, but for this cell the regulatory center is missing. That's why this nucleus of the companion cell can control or can regulate their activity. That's the one of the function of the companion cell, along with its help in the translocation. I hope that makes sense. Now let's talk about it. Now try to understand one thing. So this is the phloem, right? These are 2 different cells of the phloem tissue. Remember one thing, adjacent to the phloem you have xylem running alongside. So there can be the presence of xylem, which I have not drawn. Again, it makes no sense for the time being. Next to this, you'll be having so many epidermal cells, all those mesophyll cells who are responsible for the photosynthesis. So here somewhere the photosynthesis is going on, right? And as the photosynthesis is going on, the plant is producing glucose, the monosaccharide. Once they produce the glucose, that glucose will be immediately converted into sucrose for the transportation. Now the question is why they are not simply keeping glucose as it is. You know what happens? The moment you keep glucose here, and if you transport all the glucose, in its form, in its very own form, glucose, the glucose has a tendency to get used up. The cell may use up all the glucose the moment they are available. Not only that, glucose can also change the osmotic potential. So this can be 2 different factors which can regulate the translocation as a process. So what we can do, what if we convert the glucose into some other form which is said to be a disaccharide, sucrose, and that sucrose can be transported from one place to another place, right? Now see what happens. So this is the cell membrane of the companion cell. On the cell membrane, there are two different types of proteins that I've drawn. There can be multiple once again, but I've taken two examples for a better understanding. This protein is a type of, like carrier molecule or transport protein, which will help in transporting something. We'll see what is it. This is again a kind of protein which will help in transporting two different molecules but in the same direction, right? Needless to say, these companion cells has a number of mitochondria. So concentration of the mitochondria is more, because if you remember, I told you active transport is one of the process through which translocation will take place. It's a mass flow, right? Mass flow of the assimilates will take place. So for that to happen, you need to take ATP into consideration. So let's begin with the process, the hardcore process. Now, mitochondria. That means this is an active site of respiration. That means in this process, if you see these raw materials which are needed for the ATP, they will be utilized, and in turn ATP will be formed. The same ATP will be broken down or will be used up by this transport protein, I should say carrier protein, and this protein, with the help of the energy expenditure, will start transporting all the protons which are present in the companion cells outside the cell. So there will be an efflux of the proton molecules. So there will be an efflux of the protons, right? So I've written this H plus as a proton here. So what will happen? Now this is a classic example of active transport, right? Because you're utilizing the energy here. Now why you are doing that? The moment you send all these protons out of the companion cell, you can develop a proton gradient. That means proton will be more outside than inside. So in this way you can create a diffusion gradient, right? So now what is to be done? Since you have created the gradient, now for this proton, it becomes very easy to come back to their original place, because now down the concentration gradient they can get into the companion cell, right? Now for that to happen, the same cell membrane of the companion cell has one more different kind of protein which is said to be co-transport. Now this is a kind of typical protein which helps in the transportation of two different molecules or two different elements, but the direction of the flow will be on the same side. That is what you call as a co-transport. Now with the help of this co-transporter, whatever protons were sent earlier with the help of active transport, they will be brought back, and at the same time whatever sucrose has been made, like I already told you about, that sucrose has been prepared from the glucose, right? So whatever sucrose is present here, this sucrose will also be brought in alongside, right? So if you see, that is the purpose of sending the protons out initially, so that while they come in, they can bring the sucrose molecule inside the companion cells, right? Make sense? Now try to understand. Here you are utilizing the ATP. So against the concentration gradient. Here you are not utilizing any ATP. So this is down the concentration gradient. Sucrose is more in these cells because these are some of the cells which are producing glucose by photosynthesis. So automatically the amount of glucose will be more here. So the amount of sucrose will also be more here compared to inside. Second thing, since you have already sent all the protons out with the help of active transport, you have created a proton gradient, and that proton gradient will help the influx of the proton on its own without the expenditure of ATP. Make sense? So with the help of this co-transport, both the material will get inside the companion cells. Now if you see, these sieve tube element and companion cells, they have a tiny opening which is giving a cell-to-cell connectivity, and these openings are known as plasmodesmata. So it is through this opening now the sucrose down their concentration gradient will enter the sieve tube element. Make sense? Again, I am repeating. Initially, you had loads of sucrose molecules here because these were an active site of photosynthesis. So once these load came into the companion cell, now this companion cell is having more concentration of sucrose than this. So again, you have created a concentration gradient. So down the concentration gradient, this sucrose molecule will start getting inside this sieve tube element, right? So you see, again, this is a kind of passive transport happening. Here also you don't need to have any expenditure of ATP because you have already created a concentration gradient. Now, once these sieve tube elements are filled with, once these sieve tube elements are filled with these sucrose, that means the concentration of the sucrose will increase in these cells, as a result of which this environment will become more hypertonic because you're increasing the solute concentration. That means in this hypertonic environment, the water potential will go down, as a result of which this scenario will start drawing water from the neighboring cells. Now if you remember, I told you this side will be having xylem going alongside. So from the xylem, the water will start getting drawing in. So once you draw in the water, it will create the hydrostatic pressure. There will be a development of hydrostatic pressure. That hydrostatic pressure, the moment you develop the hydrostatic pressure here, that pressure will lead to the translocation to happen. So as a result of which, this sucrose molecule will start getting translocated towards the sink. So from the source, via different cell towards the sink, this is the mass flow hypothesis, a mass flow theory, right? That's about the translocation. I hope that makes sense to you. Now let's quickly understand, or I should say, let's quickly revise the overall process that we have discussed till now. All right. So if you see, it's starting, or what if I keep here both the page? I think that will be easy for you to understand, right? Hold on, I'll keep in this way. Right. Yeah? Okay. Active transport to pump out the proton. So we are talking about this particular part. With the help of the active transport, whatever protons were there inside the companion cells, these protons will be pumped out to the neighboring cells. Why? Just to create the proton gradient. Now, what do you mean by proton gradient? One side will be having more proton, one side will be having less proton. Since you have sent most of the protons out of the companion cell, this area will be having more number of protons than inside. So you have created a proton gradient, right? As it is created, now the second set of protein, that is a co-transporter, will come into the scenario. So this is the co-transporter. This co-transporter will help in allowing both the element, that is the proton and the sucrose, to get into it, to get into the companion cells. Because I told you it's a similar, it's a same direction. The moment they get in down the concentration gradient, that will lead to the, again one more time, formation of diffusion gradient for the sucrose inside the companion cells and this sieve tube element. So more sucrose here than this. So as a result of which, the sucrose will now start getting inside the sieve tube element via plasmodesmata. So these are your plasmod. So I have written about plasmodesmata. So these are your plasmodesmata. Make sense? Now the moment these sieve tube elements are loaded with the sucrose, that will bring a decrease in the water potential. As a result of which, water from the neighboring cell will start getting inside or getting into these cells with the help of osmosis, because you have to cross the cell membrane. The moment water is inside, the hydrostatic pressure inside the cell will increase, and that will lead to the translocation to take place. As a result of which, translocation will take place towards the sink. So if you remember, I told you in the beginning, I should say here, bidirectional transport. So sink, translocation towards the sink will take place, right? So that's it. That's about the translocation. If you have any doubt, if you have any, if you have not understood any particular part, do not forget to write in the comment section below. That's all for the day. If you have enjoyed my video, once again, do not forget to hit the like button, subscribe my channel, and share my video with your friends, right? If you have some video request, do not forget to write in the comment section below, so that I'll start preparing the content on that video, and as soon I'll be publishing a video on the same topic. That's all for the day. Take care. Bye-bye.